I am originally planned for a focus on session, and that is my first disclaimer. I am not Michel Digman. So if you expected Michel, that's not me, I'm Michael. And my second disclaimer is I will not talk about microscopy. So I will talk about single molecule spectroscopy. In principle, this is also extendable to a microscopy image. We can talk over that if you're interested in it, but today I will talk about spectroscopy mainly. And the type of spectroscopy that we use in the lab and many other labs over the world is FRET spectroscopy. And I would like to slowly, because most of you are microscopists, introduce you to what is FRET. We have heard a lot about FRET. FRET is first resonance energy transfer, or sometimes also called fluorescence resonance energy transfer. And this whole thing started about, 8 years ago with a publication by Theodor Förster. And he published a paper which is called Zwischenmolekulare Energiewanderung und Fluoreszenz. So that basically means between molecules, energy is transferred, and this has to do something also with fluorescence. And in this paper, he basically reported a derivation of a energy transfer, and he derived that also for two fluorophores. For one, fluorophore is fluorescein in solution, and the derivation basically was saying that if you have one fluorescein molecule that is excited, it can transfer its energy to a neighboring fluorescein molecule. And the second case where he derived that also where it might actually play a much bigger role in nature is for chlorophyll. So if you would have chlorophyll in solution, for instance, in principle, and in close proximity, a second chlorophyll molecule, you could transfer the energy. And what he basically found out by deriving it, looking at excitation spectra, emission spectra carefully, he figured out that if there is a distance of around 50 angstrom between these two molecules, you have a high probability of an energy transfer, and in this case of 80 angstrom. And he actually converted that also to concentration. So if you would have fluorescein in a solution of around 3.2 millimolar concentration, you would achieve these distances and you would see then this energy transfer. This is now the summary of a paper that is really old. If I ask my students in the lab, what is FRET? They explain it to me in this way. You have a donor that is highly excited, and if the donor fluorophore comes close to an acceptor, it transfers the energy and the acceptor gets highly excited, but the donor is no longer excited. So this is a student way of explaining it, but what we see also in that way of explaining it is that this seems to be distance dependent also. So what we know when we talk about FRET is that we typically know there is something happening when they are far apart. We don't get a lot of energy transfer, and these molecules are close together, we get a lot of energy transfer. So the first question that you would like to ask, of course, is what is far and what is close? And of course, this is also something that we know from literature. This is depending on a distance, but this efficiency is basically like 1 over to the power r to the power of 6 dependent. And if you look at this equation, everything comes down to this one special value in there, R0. And that is the so-called Förster radius. And this was also derived by Förster back then already. And R0 to the power of 6 is exactly this kind of equation. We had a few math things. This is not math. This is really a very, very simple equation. It depends on a few factors, and I list them here for completeness. I cannot talk about all of those in detail. One thing is the relative dipole orientation. So these fluorophores are dipoles, and if these dipoles are oriented towards each other perpendicular, there's no energy transfer. If they are parallel or parallel like that, there's a lot of energy transfer. And typically, if they are randomly oriented, we can also calculate that this kappa square parameter is about 2/3. Then there is a quantum yield of the donor in there, index of refraction of your solution, and then there is the spectral overlap integral. And this is basically the part where it depends on the fluorophores that you use. We can illustrate that with that a little bit. So the spectral overlap integral is this. We have a donor emission, here the green solid line, and we have an acceptor excitation spectrum. This is the red dashed line, and this overlap integral is this area. And if we calculate that carefully, we can then have our overlap integral and we can determine what R0 is. And this depends on the choice of fluorophores. And for most fluorophores that we use, it is typically around 4 to 7 nanometers. You can find extreme cases at the lower end that go even down to 2 nanometers or less, and you can find extreme cases at the higher end, of course. So what we note is basically the efficiency of energy transfer is distance dependent, and this is a non-radiative energy transfer. So we have a dipole-dipole interaction, which leads also that we have this R to the power of 6 dependency. So it's not that the donor is emitting a photon and the acceptor catches it. That would be a super low probability, actually. So let's look at that equation a little bit more detailed. If we plot this, we see this line here, the green line here, and that is something that everybody would know. And of course, what people came up with when they saw this is like, oh, we measure an efficiency. And when we measure that efficiency, we can derive a distance. Now, this example here, for instance, when we measure 80% efficiency, we have a distance of 4.7 nanometers, for instance. Or if we measure an efficiency of 30%, we have a distance of 6.8 nanometers. So you see this is a highly sensitive technique also. Like 30% efficiency versus 80% efficiency is something that in principle we should be able to determine. And there the length change is only about 2 nanometers. So we have most likely also angstrom resolution in between here. And that is, this is another historic case, is why FRET or energy transfer was termed as a spectroscopic ruler. And one thing is there, of course, these are also the length scales of proteins, as Ralph also explained to us already, that proteins are on the size of a few nanometers, and therefore this would be perfect to also look at intramolecular motions within a single protein, because we are also sensitive to that. So my molecular ruler is this. We have a ruler, we have a donor and an acceptor, and we measure an efficiency, which is nothing else than a probability to transfer the energy from a donor molecule to an acceptor molecule. So we can ask, of course, the question, how do we actually now how can we actually determine this efficiency? Like I always say, like we measure an efficiency. So how can we determine a FRET efficiency? And there are two fairly simple ways. One is called the intensity-based way. So I expand this equation up there, and the FRET efficiency is also nothing else than the intensity of the acceptor divided by the intensity of the acceptor plus the intensity of the donor. The other way would be a time-resolved way that is given by this equation. So that is depending on the fluorescence lifetime of the donor in proximity of the acceptor divided by the fluorescence lifetime of the donor in absence of an acceptor. And why is that the case? I quickly like to illustrate that here with a Jablonski diagram. So this is a diagram of the donor fluorophore. We excite it, and once the electron is in the excited state, it can go different ways. The easiest is it jumps down and emits a photon, that's fluorescence. But there is also internal conversion, intersystem crossing. There might be even many other ways. And if we want to calculate now the lifetime of this donor molecule, we basically have the rates for all of these three transitions here, radiative, non-radiative, intersystem crossing, for instance. If you have an acceptor in proximity, you have another exit here for that electron, and that exit is basically it donates its energy to an electron in the acceptor, so to speak, simplified speaking, and then the acceptor electron is excited and we observe something, but we have a new exit path, and this transfer rate here, kT, is again R to the power of 6 dependent. That's why we can use this simplified equation as well. So now we know we can measure a FRET efficiency when we measure intensities or lifetimes, and we can then maybe calculate a distance or a molecular distance. And the field that was developed about nearly 30 years ago was thinking maybe 10 years ago about the question, how accurate and precise can we actually do single molecular FRET measurements? So if you want to do accurate and precise measurements, you need a reference system, and the best reference system that we know, we learned about that, is DNA. We have millions of crystal structures. We have a very good understanding of DNA structure. So let's use DNA there. And the idea was we take double-stranded DNA and we place a donor and an acceptor molecule at different bases. We can model the fluorophore accessible volume. And then we basically find out when we do these experiments that the uncertainty for the length measurement is about 1 to 2 angstrom, less than 5% of the distance that we measured. And this is not only we, but this was a huge study of an entire field where more than 20 labs in the world came together to really determine that we can measure accurate and precise distances. This was published in here, but one of the big things that actually came out from the study is that the field came together and defined standards on how to analyze the data. That led to slightly ugly equations. This simple equation with an intensity and 2 intensities out there came out to be an equation a little bit longer like that. We have tons of correction factors that are instrument dependent, fluorophore dependent, detector dependent. So we basically have to take all this into account, but if we do, we can be very accurate and precise. The field was so excited about it that actually a couple of years later, we thought well, let's take not DNA, it's a little bit boring, let's take a protein. And we can do that also on proteins. And there again, we can be fairly precise, less than 2 angstrom compared to a crystal structure, fairly accurate. But the most important part there of that publication in my eyes is that we actually also can see dynamics and basically see breathing of that protein and motion of the protein. So now we know we are accurate, we are precise. You can use it if you want it. You don't have to use it. You can also just look at changes. But how do you actually do single molecule FRET experiments? And there are 2 general approaches on how to do single molecule FRET approaches, or FRET experiments. And the first one is you immobilize your molecules of interest. I depict here in a very simple cartoon, double-stranded DNA, a donor and an acceptor molecule. And then once they're immobilized, you can somehow excite them and look at donor fluorescence and acceptor fluorescence. Now, one way to do that is you take a very simple turf microscope. That's a home-built instrument, for instance, but there are also commercial instruments that work like that. In this case, this is a prism-type turf instrument. You record that, you split up the donor and the acceptor intensity, and you image that on an EMCCD or a CMOS camera. And then what you see is typically something like that. We have here the donor signal, we have here the acceptor signal, and you see individual molecules here. And from that, you can directly read out, for instance, the intensities and then calculate FRET efficiencies. So these intensities over time will look like that, for instance, green and red here over time, fluctuating here in this case because we have a molecule that is actually changing its structure over time. And then you can calculate out of these intensities the FRET efficiencies, and you can then monitor the FRET efficiency over time. And this is not a bad signal quality. I want to point out that this is intentionally a molecule that fluctuates between different states, a protein that assumes different kind of conformational states in this case. And by doing these kind of experiments, we typically obtain these time traces with transitions between different states, which we can then model later on using hidden Markov models and many other methodologies that have been developed over the past 20 years. And we can observe single molecules between 10 and 1,000 seconds, depending on your illumination, on your time resolution, and so on. But basically, it means we can monitor a single protein and its conformation over about 1,000 seconds. And of course, you can do that with 5 or in a normal field of view with hundreds of molecules in parallel. The other approach is the following. I like to depict that here. For instance, if you have a protein which you cannot easily immobilize on a surface, you would like to measure that in solution, and this protein undergoes also some kind of a conformational change, then you can think of it as well like you try to monitor that with FRET. So you attach donor and acceptor fluorophores at the end of the protein, for instance, and then you do an experiment in a confocal type single molecule FRET instrument, where you basically focus the excitation light and then detect from a confocal volume. You dilute your sample to a very low concentration of picomolar in this case, And then these molecules will occasionally diffuse through your confocal volume, and you record acceptor photons and donor photons, these so-called burst traces here on the top right-hand side. Yeah. So you see that occasionally the fluorescence goes up, and this was always the case when a molecule was in your confocal volume. If you then go to one of these bursts, you can collect 4 acceptor photons, whatever tens of donor photons, or other cases, and third case where you get more acceptor photons, for instance. And then you can calculate again from these photons, using this equation, the efficiency of all these 3 molecules, and you can put them in a so-called FRET efficiency histogram, and you do that not only for 3 molecules, but you wait a little bit longer, and then you can obtain a so-called FRET efficiency distribution, for instance. From that, what you read out already is that you obviously don't have a homogeneous population of your protein structures, but a heterogeneous one with at least 3 different kind of states, which you could approximate, for instance, with 3 Gaussians here in this case. So all of you might be hungry, so I have a funny slide in between. So why do you want to do FRET? And I answer this question from my perspective. And I would like to start out with a academic family tree. So there was Theodor Förster, and he had a PhD student, and one of his PhD students was Eric Sackmann. And Eric Sackmann had another PhD student, and that was Hermann Gaup. He had a PhD student that was Matthias Rief, and actually Matthias was my doctor father, so I was a PhD student of Matthias. So the fun fact is I'm a great-great-great-son of Förster. That could be a motivation for doing FRET. Actually, I figured that out much later, and most interestingly, none of the people in between here did FRET experiments. So why do you want to do single molecule FRET? And this is a little bit more the motivation why we do that in our lab. We use single molecule FRET because it matches the length scales and time scales of biomolecules. So things happen on a millisecond, microsecond time scale. It allows us to measure structural changes. It allows us to measure dynamics. And it allows us to actually decipher molecular heterogeneities that we typically observe in nature. Some of my students also just like to watch this in a dark room because it feels a little bit mesmerizing. So if we now want to do actual single molecule FRET experiments, the first question is, how do you attach 2 fluorophores to your molecule of interest? You would take here the protein, undergoes this conformational change, you would like to probe it. So you need to attach a donor and an acceptor. And we heard beautiful talks using also FRET-based sensors that use fluorescent proteins. I make a simplified sketch of that. This should be like the donor molecule, this should be the acceptor molecule. And that's a great idea for proteins. Yeah, it's genetically encoded, you could express that. So you have a high labeling efficiency. That's the advantage. The challenge is they are a little bit bulky. Fluorescent proteins are about 27 kilodalton or 4 by 4 by 4 nanometer cubic. It does not work for DNA and RNA. So if you're interested in RNA dynamics, RNA structure, DNA dynamics, and they have a very limited photon budget. Quantum yield might be buffer dependent. There are tons of different fluorescent proteins, and sometimes they help, but for looking at small conformational changes, they were not that helpful for us. So how do you attach that then to a protein? Idea 2 is you attach 2 organic fluorophores using a chemical reaction, and I briefly would like to explain that. So you attach it to N and C terminus, and they are much smaller, and for simplification I just illustrate them here either as dots or as stars. This has nothing to do with one or the other reaction site. I just want to say throughout the talk, could be dots or stars. And there are different methods on how to do that, and I don't want to go into all the chemical details, but this is something that we deal with every day in our lab. And the easiest way is you introduce cysteines to your protein of interest, and then you use a Maleimide-Michael addition reaction. This is one of our favorite approaches, not because of the Michael, but this is a very simple reaction. This is a reaction scheme here. So you can throw things together, wait a little bit, and then you purify it, and then you have fluorophores typically on your protein. If that does not work, for instance, your protein might have 20 native cysteines, and you do not want to remove these cysteines. You have to come up with other ways. And one way that we also use in the lab is you start protein engineering. That means you introduce, using amber codon suppression, a chemistry group with unnatural amino acids that allows you, for instance, then click chemistry approaches, as illustrated down here, with alkynes and acids, for instance. And a third approach that turned out to be also very useful in the lab is you use tags and conjugating enzymes. That means you can encode short peptide tags into your protein of interest, and then you use a chemical reaction, in this case mediated, for instance, by SFP, that attaches coenzyme A. And coenzyme A luckily has a thiol group, which you can label beforehand, actually, already, with your fluorophore. Then you have coenzyme on your fluorophore in solution, SFP, and then it attaches it to that serine residue. And this works surprisingly efficiently in our lab. So we get labeling efficiencies of 60 to 70% using this approach. That works great for a protein. Of course, we ask ourselves also, how does this work for nucleic acids? For DNA labeling, this is super easy. You go to a company and you order your labeled oligo. Costs you a little bit of money, but you can order it either with your fluorophore or with an amino modified base. RNA labeling, which has become of interest in our lab over the past couple of years, is not so easy. So you can order short RNA labeled, but this is already slightly more expensive, about a factor of 5, 6, 7. But then with long RNA, and long means already something above 40 nucleotides, this becomes really difficult, and we start to do that in the lab. And I briefly want to mention 3 cases. 3' labeling works great. There's T4 RNA ligase. There are fluorophores conjugates available that you can buy commercially, and that T4 RNA ligase very efficiently labels the 3' end. On the 5' end, if you look in literature, you find tons of protocols and tons of approaches, and I have a very talented biochemist in the lab. And always the yield was in the 5 to 8% range. So we sort of like said, we gave up on 5' labeling for the moment. If someone has an idea, I'm happy to listen. But we came up also with an idea on how to do internal RNA labeling, which gets much more interesting if you have long RNA. And that was inspired by a great paper by Claudia Hörbartner. And I would like to point that out. So she uses a ribosome. That's this ribosome here. And if this binds to your RNA and there is a adenine residue, you can supply in solution, for instance, M6 guanine. It takes the methyl group and it transfers the methyl group to that adenine, and then you end up with methylated adenine here and guanine somewhere in solution. So all done by an RNA enzyme here. So interestingly, you have commercially available SNAPtag ligands that carry on the guanine not a methyl group, but some kind of a group and a fluorophore. And if you throw that in, that ribosome doesn't care if you have a fluorophore there and not a methyl group. It transfers exactly that fluorophore and this group here to your target, and you get a labeling efficiency of around 60 to 70% as well. And this works obviously with a lot of SNAPtag ligands that are commercially available, but we are also like at the moment exploring options of making our own fluorophore conjugates with this kind of a start product here. So now we know how to attach fluorophores to our structure of interest, might it be DNA, RNA, or a protein. Now we have another problem that I have not talked about, and that is the problem of 2 fluorophores. So you have labeling efficiencies for 1 fluorophore, you have labeling efficiencies for 2 fluorophores. If you use 2 cysteines, your protein of interest, you can end up with this structure. You can end up with that structure. For a distance measurement, it should not make a difference, and I'm happy to talk about that, why I say should, because in some cases, unfortunately, it does, and we have to control that. But what we find also in solution is, of course, this and that. So now we have a pool of all kind of species, and you could do some ensemble thread experiments in your cuvette, but then you basically would have a lot of contamination in there, so to speak. So something that doesn't give you a signal. So it would be nice if we take this pool here and if we could sort it out. If we can say like molecules that carry only donor fluorophores go here, molecules that carry only acceptor fluorophores go there. And this is something that the field also came up with solutions a couple of years ago. The Kapanidis and Weiss lab, or the Lamb and Seidel lab, they came up with a technique which is called alternating laser excitation or pulse interleaf excitation. And the idea is very simple. Instead of just exciting your donor fluorophore, very briefly in between, you directly excite your acceptor fluorophore to test if that molecule that you're probing at the moment also carries an acceptor. Why is that interesting? Well, we can define a new parameter, which I call here, or the field calls stoichiometry. And that looks a little bit like a Fred equation, but it's actually like the number of photons of the acceptor after donor excitation plus the number of photons of the donor after donor excitation, and so on and so forth. And there's one important parameter, that's the number of photons of the acceptor after acceptor excitation. And if you look carefully at this equation, it turns out that S equals 0 when you carry only acceptors, S equals 1 when you carry only donors, and S is around 0.5 when you carry a donor and an acceptor molecule. And then this looks like that. So you plot a stoichiometry versus a Fred efficiency. And each of these dots is a single molecule that was diffusing through a confocal volume in this case. And you can then basically like say we have stoichiometry versus Fred efficiency and applying some kind of filters, and I'm happy to discuss also these filters offline. You can select molecules that are molecules that have for sure one donor, one acceptor molecule. And this helps you to have not only a Fred efficiency histogram as you would normally get, but you can actually filter out those that really carry a donor and acceptor, and you obtain then a Fred efficiency histogram that reflects your molecular conformation or conformational example. So I show here data from a confocal instrument, but in the lab we actually also operate a turf Fred instrument with alternating laser excitation, and they have both their advantages and disadvantages. Here we get time resolutions of 20 millisecond to minutes. Here we get nanosecond to millisecond time resolution. Here we can do a lot more about fluorescence lifetime, anisotropy, and so on and so forth. Here we can record for thousands of seconds, and this is a fairly simple instrument. So I was invited here to talk a little bit about high throughput methods, how we can use Fred spectroscopy to help us with drug discovery. And I start out with high throughput. And in the past 2 years, there have been 2 developments in that field. And I would like to briefly introduce both developments. One by our lab, that is part 2. And the other one was actually like in parallel by the Chilmin Zhou lab in Delft and the Sebastian Deindl lab in Sweden. And they developed a method that allows them to do also high throughput total internal reflectance fluorescence resonance energy transfer experiments. They called their method Sparks and Muscle. And I would like to briefly talk about this. Obviously, I'm not the expert and I cannot give you any insights of all the tricks and details, but I would like to introduce you what the concept and the idea behind that is. And both of these labs were interested in DNA sequences and how DNA sequences affect, for instance, DNA structure, DNA dynamics. So when you think of DNA, most of you might think also of sequencing, and that's also how they started. And what they discovered or thought of is that next generation sequencing is already fluorescence based. That means if we take an Illumina chip, we might have already a high throughput instrument that allows us to do high throughput measurements. So this is a brief scheme of how a Illumina chip might look like. So you have a surface where you can immobilize your DNA that you would like to sequence. You amplify it, and then you have your 4 nucleotides swimming through the solution with fluorophores attached. and then you just read out which color was attached to that, and you basically, by that, reconstruct the sequence of your original DNA strand. Though I would like to point out, this chip and this methodology is not single molecule based. It typically has hundreds to thousands of strands there that carry maybe, hopefully, all the same kind of fluorophore, and this is a signal that you can then easily read out. So how do you transfer that to a single molecule technology? So over the next 2 slides, I briefly explain that and give you an idea on that. These are very similar, and both are building on the same Illumina chip. And the concepts are very, very similar when you read both papers that came out last year. You see that, yeah, they are independent, but also not so independent if you think about what they designed. So I show interchangeable figures from one or the other paper. So the True Lab, in this case, was interested in a DNA sequence, which is called a holiday junction. That's one of the intermediate elements during a DNA recombination. And they were interested in if all bases or some bases close to that central cruciform-like structure influence the dynamics and the kinetics and the overall population of a low Fred efficiency state or a high Fred efficiency state. So the way how they designed now their experiment is the following. They generate a long piece of single-stranded DNA that would form, in this case, in a holiday junction-like structure attached to fluorophores, and this holiday junction will either assume this state or that state. This is transient, it never exists really, but it helps us to understand a little bit the sequence diversity. They made these strands mostly complementary, and then they kept 8 positions down here where they incorporated through random gene or DNA synthesis, in this case, random nucleotides. So overall, they would expect that they get 4,000 different DNA sequences. These were then diluted and immobilized on this Illumina chip. And then you can immobilize them through an overhang sequence to a complementary sequence that is anyhow present on these chips. And then you have them present there. Then the workflow is you do a single molecule experiment. So you take the chip, move it to your custom modified TAR FRET instrument, you image. Then afterwards, after the imaging, you move the chip to your Illumina sequencing facility, where they actually amplify your original sequence so that you have thousands of strands, and then you do sequencing. And then afterwards, you can correlate that random sequence with what you observed in your single molecule FRET experiment. So that sounds quite exciting. If we look at the numbers that they give in the papers, this looks intense. For instance, in that paper where they studied the holiday junction, they were imaging for 5 days. They detected about 10 million molecules. That looks like high throughput. Then they did sequencing in parallel. In sequencing, they actually could only identify about 2.7 million molecules with the sequence. And through some selection criteria that they have, they ended up from, in the beginning, 10 million molecules, around 400,000 molecules. That's still impressive, very impressive for me, because when I do my TURF FRET experiment, I observe typically 400 molecules. It's a factor of 1,000. And things that you can then correlate after these experiments, and this is like where I jump to a very different biological question, but I still would like to show what they correlate. They basically observe for one molecule a FRET efficiency distribution, for instance, and then they can correlate this distribution with the sequence afterwards. So they have a random sequence again, and they can correlate that. Or for instance, they can correlate, if you are rather in the high Fred state, you can correlate that with a different sequence. And by that, you can learn something about your underlying sequence diversity. In this case, about Cas9 finding a target, for instance. And this workflow is potentially to be expanded for 10,000 different sequences or even more. But, and this is my point of view, it is heavily DNA based. So it is basically requiring that you look at DNA and DNA sequence variations. And then the second part is these are highly customized instruments. So you basically like have to use or modify your instrument specially for this kind of purpose. On the other hand, you can really like then tackle questions like what kind of DNA sequences might be important for histone positioning and nucleosome formations and so on and so forth. And these are like some of the visions that they discuss also in their publications. So now over the past over the next 10 minutes, I would like to talk a little bit about our development in the direction of high throughput single molecule FRET experiments. And this basically uses multi-parameter fluorescence and time-correlated single photon counting. But the big question that we have here is why did we actually develop this large throughput single molecule FRET workflow? And I would like to give you a little bit of a background there. And this all started with a master thesis actually of a very talented student in the lab, Antoine Treff. Antoine was interested in misfolding of a protein. In this case, the protein is called CFTR. And what was known is if you have a point mutation in CFTR, this causes a disease called cystic fibrosis. Some of these point mutations are also like in transmembrane regions. And for instance, here in the region of the transmembrane helix 3 and 4. And when he started his master thesis, we said like, well, we cannot express and reconstitute this full-length protein. Back then, it was basically not possible to easily express it. So we can take a subsection of the protein, just these transmembrane helixes, and like to look at it, how can they potentially misfold with a patient-derived mutation? So Antoine attached to that tiny little hairpin, which he expressed and purified, a donor and acceptor fluorophore, and then he reconstituted everything into LUVs. This is not to scale here. Yeah? These are 100 nanometer vesicles, 4 nanometer thick, so roughly not to scale. And then he put it into our confocal instrument, and they were diffusing through, and we could read out a FRET efficiency histogram. And what he did there is 2 experiments. Well, in the end, 2 experiments. He did 100 more. But what I would like to show are 2 FRET efficiency histograms, one of the wild-type protein and one of a patient-derived mutation, V232D. And you see that this is very different than this FRET efficiency histogram. And by doing a few more experiments, we can derive actually that this is nicely inserted. A majority of the protein is perfectly inserted. Some seem to be less inserted. And if you have the patient-derived mutation, you actually have a larger distance, when we can actually deduce that one of the alpha helices is in the interfacial region and not completely inserted. That was very nice, but Antoine didn't stop there. He found a small molecule that was reported in literature to be developed for the treatment of cystic fibrosis, not for this mutation. But he said like, oh, it is a very lipophilic drug. Let's try to throw it on these vesicles with the patient-derived mutation, and he titrated it in with larger and larger concentrations, and he could actually like refold the misfolded hairpin structure to a folded like hairpin structure, where you have then a largely predominant population of molecules that are correctly folded. And that was super exciting for me, also for Antoine, for the entire lab, because that said like, well, we have now with FRET a very simple readout. We don't even need to measure the exact distance here in this case. This is a readout, am I in state 1, correctly inserted, or in state 2, incorrectly inserted. So here's the problem. We have more than 500 known mutations in these transmembrane regions. We have 5 approved drugs. There are hundreds of small molecules that are at the moment in development for exactly cystic fibrosis. If you then do combinatorials of this, it turned out that I probably could have hired 5,000 more master students to do these experiments and supervise 5,000 more master theses. Obviously, that's not the way to go. And that's when 2 people in my lab teamed up that were more from the physics side and from the engineering side, Kushik and Andreas. And they developed our approach, which we call automated multi-well single molecule FRET microscopy or spectroscopy. This little GIF here already illustrates the kind of workflow. So we basically use pi interleaved excitation, pulse interleaved excitation. We excite all these molecules. We scan through well by well, and each well has maybe a small different molecule or a different point mutation. So this was the idea and the motivation. Obviously, motivation is good, but doing it took us a little bit longer. The simplest case is like, well, we implement in our system a large distance scanner that allows us multiple plate reading. Very standard. But then we faced the first problem, sample evaporation control. Each of these samples is about 100 microliters. You can cover this up, but still you might have some sample evaporation problems, especially if you want to control your concentration. If you go from 100 microliter to 80 microliter, your concentration is different. If you go to 10 microliters, it's a lot different. The trick is published. It's very, very simple. You do the PCR trick. You heat the surface a little bit more than your solution. So we put here a pad that is sold on Amazon for aquariums where you can heat. It's 10 euros. You put it on top and you put it 1 Kelvin warmer than the room temperature, and you don't get condensation on the top. And you can seal it up with a parafilm beforehand, and then everything is fine. But it took Kuschik actually a few trials to figure that simple solution out. We have to control immersion water evaporation, but there are commercial versions for that. There are also like other ideas that we are currently testing. But the biggest part was the software development, because we do not do one experiment, but we do 96 experiments. And we have to treat the data, analyze the data, pre-analyze the data on the fly ideally, and then also visualize that. And I will talk about the software at the end briefly. So the first thing was, of course, are we accurate, precise? How stable are we? Very simple experiment. We take the same 2 rulers that we used in this very first Nature Methods paper, where the field came together to test precision and accuracy. But we now put these rulers into 96 wells, always together. And then we wanted to find out, well, if we start our experiment at well A1 and we end up at H12, how comparable are the data sets? So we measure per well 20 minutes in this case. And then I show you some examples, well 15, 45, and 75, and we get these thread efficiency histograms, and they look fairly similar. What I could do now is I could show you 96 thread efficiency histograms. This might be a little bit boring also for us. That's why we came up with a different visualization. We actually like visualize this now by a color heat map, and each line here is a single well. And in this case, we have 96 times the same sample. We measure the thread efficiencies, and we can actually find out that the mean absolute deviation is less than 1 angstrom from well to well, which was quite impressive. And what was more impressive is that when we start our experiments, we typically in these 20 minutes acquire 1,500 molecules. When we end our experiments, we typically acquire around 1,500 molecules. And this is over a time course of 32 hours. That's still a very long experiment, but it is fully automated, so nobody has to sit there, nobody has to exchange solution every 30 minutes. Tough student. But in this case, it basically means we don't have any evaporation problem control, and we can measure for 32 hours with a stable instrument. And I don't show you all the experiments that are described in the publication. I would like to point out a few. So we were also interested in if we can measure kinetics with that on a molecular timescale. And the simplest system that we use there is a DNA hairpin-like structure that carries a donor and an acceptor fluorophore. And it anneals with 5 base pairs here in this case. So it is very transiently, and we can influence that with salt, as we also learned in the previous talk. If we do these experiments and we find a pipetting robot that can take us through 96 different salt concentrations, increasing from 100 millimolar to about a molar in sodium chloride in this case, we can see that the thread efficiency histograms change from rather low thread efficiency to mid-thread or 2 populations at the highest salt concentration. We can then look at these thread efficiency histograms. Unfortunately, they are sorted here from low salt to high salt. This is the other way around, so I flip it for you. So we have here high salt down there, we see 2 populations, and then we see also in between a lot of molecules, and these molecules are dynamic, and I briefly would like to illustrate that. If you have a molecule diffusing through your confocal volume, and it decides in between to change its conformation, the average thread efficiency will be somewhere in between. This looks very different if you use something that is very slowly and rarely changing the conformation, for instance, a protein. This is also shown in the paper. This doesn't change typically its conformation within the observation time, and then the thread efficiency has these two distinct peaks. So this is the peak that carries the dynamic information, which you can then use to extract for each salt concentration some assembly rate and disassembly rate, closing and opening rates. What was more interesting in that experiment is we can also define confidence intervals. So if you do these titration experiments by hand, you typically do maybe 5 different concentrations, so you have still a large confidence interval. If you do 20 data points, that is probably sufficient. You have a 5% confidence interval in this case. You can now build up things very complicated. You can look at 3 component systems, and I see I have a few minutes left only. So if you do 3 component systems, you can ask a lot of questions on how 2 proteins, in this case SSB and RegA, compete for the same single-stranded DNA. So you can make titrations of both together. This is how the data looks. I will not go into all the details, but what I can show you is we can extract now for an increasing SSB concentration at a constant RegA concentration, the change of fraction of molecules being bound by SSB, for instance, and we can then actually build up a thermodynamic equilibrium model and extract information about how SSB stimulates RegA filament formation. That actually this increases the affinity and it reduces the Hill coefficient of filament formation for RegA. This all comes with a software. We have PyMulti, PyBat, and PyVisualization. These 3 pieces allow you to actually interface to commercial existing systems. You can then automatically process all these bursts of all these 96 wells. It keeps a folder structure carefully, and you can visualize single well data next to 96 wells in parallel. So I would like to come back to the beginning of the story. This is the work of a PhD student, Matthias Schenkel, in the lab. He then generated a library of patient-derived mutations, and he titrated in different kind of small molecules. So depending on the patient-derived mutation, we can now use this assay to first of all find out if we have misfolding or not in the patient, most likely. And if we can find a small molecule that by titrating it in, we can correct the folding. We can do that for different molecules, and I show only 3 here, where we can actually find that they are depending on the mutation active. And we have also the collaborators in Italy that develop a new molecule that shows activity also to 2 other mutations. So with that, we have, of course, now the possibility to generate larger libraries of more patient-derived mutations of all kind of transmembrane helices in this case, and find out what is the mode of action of these small molecules. We can think of serotyping. We can screen new compounds, specific and non-specific corrections of pharmacochaperones. And of course, this is something that can be expanded to many other misfolding diseases of transmembrane proteins, for instance. How many minutes do I have? 2? I negotiate 2 minutes. So 2 minutes, just conceptually, what can you use now this system for? I would briefly like to say microRNA is one of the interesting targets for the lab at the moment. MicroRNA is very important to regulate processes, but it has also raised interest as being a biomarker for instance, cancer, diagnosis, treatment selection, or prognosis. There are tons of different microRNAs that are also involved in different diseases. I just show you some examples here in this case. So we thought let's develop a microRNA sensor that we can actually help maybe with the diagnosis platform, because there are on the current systems a few different problems. And the system is very simple. We take a hairpin. The hairpin opens and closes, and then we encode here a sequence in the hairpin that is complementary to the microRNA, and then we basically kinetically lock the hairpin in a low thread state in this case, for instance. So the first target that we tried on is microRNA 184, and this is something that we screened with this 96 well plate assay, where we then actually designed different DNA variants and used also different microRNAs. And if you then titrate in, for instance, the microRNA to these sensors, you see they end up all at the same state. but they show all slightly different affinities to the microRNA. This is all done in a single measurement in a single 96-well plate experiment, for instance. Now we can also introduce point mutants in the microRNA, and you see they bind differently. And actually, when you do the full titration curves, you see also that, for instance, for this sensor here in the center, we can discriminate a single base pair nucleotide difference here with a dramatic change in affinity, for instance. I hope that I convinced you that FRET, and especially multi-well FRET, is pretty cool. We achieved a few different things. The software is completely open source, so you can download everything if you're interested in that and implementing it in your lab. I would like to thank collaborators funding, but outmost I would like to thank all the different team members over the past 10 years that were involved in actually developing these things in our lab. If you want to be part of that, we have open PhD position in my lab 1, but we have a research training group recently funded, and we have 12 open PhD students positions, and the application deadline is October 24. Not sure if I've written that somewhere here. Just look at Dresden condensates if you're interested in biomolecular condensates, because that's where it comes to imaging. You can image now molecular structures on a single molecule level in a condensate. And if you're interested in doing that, just apply here and join our lab. Thank you for your attention.